Well, hello everyone. I'm Guillermina Canesini. I work at Instituto de Salud y Ambiente del Litoral in Santa Fe. I want to welcome everyone for this symposium. It's a pleasure to stay here, and I want to thank to the Sociedad Argentina de Investigación en Neurociencias for allowing us to be a part of this annual meeting. Well, this symposium is titled Perspective in Research of Behaviors Associated to Food Intake, Memory Impairment, and Sleep Restrictions. And it's known that the alarming increase of overfeeding and the associated metabolic and immunological disorders make the etiology and the consequence of obesity a widely studied topic today, and receive increased attention of the scientific community. The goals of this symposium is present published and unpublished results or findings about the behaviors associated to food intake, memory impairment, and sleep restrictions in rodent and zebrafish models. Well, for that, I need to introduce our three speakers. First of all, I have the honor of present our captain on board. Yesterday we canceled her, and Dr. Maria Florencia Rossetti. If you can share your screen, Flor. Well, she today cannot be with us in person, but we can learn about his work about neonatal overfeeding, implication of hippocampal dysfunction, and the environmental intervention as a mitigating factors. Well, Flor, it's your time.
Thank you so much, Lisha, for the presentation. Can you listen me? Okay, it's okay?
Perfect.
Okay. So, well, hello everyone, and thank you for the presentation. Thank you, Guillermina, for this invitation, and also thank you to the audience for being here, and to the Sociedad Argentina de Investigación en Neurociencias for allowing us to be part of this Congress. So, as you probably know, obesity is one of the most public important challenges in the world, since it has several social, emotional, and physiological impact in the people. This is some information from the World Health Organization. In 2016, more than 1.9 billion of adults were overweight and 30% were obese. But most important, more than 300 million of children and adolescents were overweight or obese. And in 2020, this value was 39 million of children under the age of 5 were already overweight or obese. So there is an increasing interest in understand what is caused, what are the mechanisms that are involved in obesity, and which are the consequences of this pathology. Obesity is really important because it decreases the quality of life and has another implications like metabolic, hepatic, cardiovascular disorders. And during the last year, there are also growing evidence, epidemiological evidence, that show that an increase in adiposity could cause an increase in the risk of some neurodegenerative disease like Alzheimer's disease and other kind of dementia. So, as I mentioned, the mechanisms that are involved in these two conditions have been studied for more than a decade, and some authors propose that metabolic disorders exacerbate neurodegeneration and neuroinflammation. More important is the obesity in children, since children are at higher risk than adults because their neuronal and physiological systems are still developing. In fact, there is a growing interest in knowing how obesity during early postnatal development could have impact later in life and produce another conditions like obesity, addictive disorders, and dementia. So based on all of this, we start working in the lab with a neonatal obesity model because we wonder how overfeeding during early postnatal development that causes obesity during this early development could affect metabolism and behavior. So for that, we use an experimental model that is called small litter, and is a model well established to study neonatal overfeeding and neonatal overweight. In this model, we reduce the litter to 4 pups per mother. So the pups could ingest more milk and gain more body weight than normal litter that in general are 10, 12 pups per mother. So we reduce the litter at postnatal day 4, and to characterize the model, we study the effect at postnatal day 21 as a short-term effect and postnatal day 90 as a long-term effect. All the results that I will show you today are were done in male rats. We are also working with female rats, but the results are not ready to show. So what we found, well, as I mentioned, we performed the reduction at postnatal day 4. So at postnatal day 7, we already see an increase of the body weight in a small litter compared to normal litter, and this increase is maintained until postnatal day 21. And we also found some changes in metabolic parameters. Small litter animals has more weight in the epidermal adipose tissue weight is higher in small litter animals, and also there are an increase of some metabolites like cholesterol, glucose, and triglycerides. When we study this effect in adult animals at postnatal day 90, we see, well, respective to the body weight, we found that the increase in body weight that we found during neonatal period was maintained until postnatal day 80, and at postnatal day 90, in the moment of the sacrifice, the difference in the body weight was not found. So the animals restore the change in the body weight. We didn't find difference in these animals in terms of metabolites, epidermal adipose tissue, and food intake. So we wonder how this effect that we found during neonatal period could affect behavior in the adult animals. So we performed 3 behavioral tests. The first one is the locomotion activity test. I am not sure if you are familiarized with this kind of test, but we leave the animal in the center of the open field, and we leave the animal to explore the open field freely during 10 minutes. And we analyze some parameters. Here are some of them. For example, the distance traveled, there were no differences. Also, the time in the outer zone that is related with an anxiety behavior, the time in the inner zone, and the defecation. So what we found here is that the small litter increased the time in the outer zone, reduced the time in the inner zone, and also increased the times of defecation. So this means that small litter could be more anxious than normal litters. To prove that, we perform a test that is specifically for anxiety-like behavior that is called the elevated glutamate test. And this, I put a video to show you. So here, we leave the animal in the center of the maze, and we leave between 5 and 10 minutes to explore the 2 arms, the closed arms and the open arms. The open arms have an anxiolytic effect. So if animals spend more time in the open arm, we expect them to be less anxious. So we analyze the time of the animal enter to the arms, the time that they spend in each arm, the time in the center, the distance travel, different parameters. So we found particularly that the small litter expend less time in the open arms compared to normal litter. So this means that these animals could be more anxious than normal litters. So finally, we perform the episodic like memory test. Episodic memory is the memory of the what, where, and when. And we can analyze the kind of memory in the animals using a combination of object recognition tests that analyze the time that animals spend with each object that were presented in different moments and different positions. So there are 2 trials in which the animals live in the open field and explore different objects, and the test, all of them in the same day. So basically what we analyze here is the time that animals spend with each object. 2 objects for the first trial, 2 objects for the second trial, that we call old object for the first one and new object, recent object for the second. And also the objects are in a new position that were not presented before, that we call display object, and in a position that was presented before in the both trial, that we call a stationary object. This is just a briefly explanation that how the test is, but basically what we found is that normal litter has a good performance in this test. We expect that animals prefer to spend more time with the old object related with the recent object and with the display object related to the stationary object, and that was the pattern of the normal litter. But when we analyze a small litter, if you see in the picture, these are the different objects that we analyze. There are no preference for the object. This means that these animals could have alterations in the episodic memory. And this is also shown here when we analyze the ratio of temporal component and spatial component. So if we see normal litter has positive values in temporal and spatial component, while a small litter has values near 0 or negative. So this means that this component, that are the 2 components of this memory, are there. So up to now, as a briefly summary, we see that litter size reduction induced metabolic alteration at postnatal age 21 that disappear at postnatal age 90, but in adult animals, we found that litter size reduction promotes anxiety-like behavior and a poor performance in episodic late memory tests. So this makes us our second question, if the environment could reverse this effect, the kind of environment. And for that, I want to introduce enriched environment. Enriched environment is defined as a combination of complex inanimate and social stimulation, and it has been used during several years to analyze the effect because it was reported by several authors that enriched environment has positive effect on brain functions, cognitive functions, neurogenesis, synaptic plasticity. It increased the expression of neuroprotective factors, and also it's really useful in neurodegenerative models like Alzheimer's disease and Parkinson's disease, because it was shown that it covered the cognitive abilities and motor abilities and cognitive functions. So basically, in the enriched environment, animals are housed in bigger cages than a standard environment, with a lot of animals to promote social stimulation, and we include different objects of different size, shapes, and colors to promote curiosity and exploration behavior, and also running wheels to promote motor activities. So what we do after that, so we work with small litter and normal litter, as I mentioned, and at postnatal age 21, we house them in a standard environment, like here in the picture we can see. There are no stimulation, we leave 4 animals per cages, or enriched environment. I also include a video here, so bigger cages are bigger. We include more animals between 8 and 16, and we leave a lot of objects to promote curiosity and exploration. And at postnatal age 90, we analyze different parameters. What we find in this after this stimulation, so environmental enrichment decreased the body weight in both groups, small litter and normal litter, from postnatal age 28 to postnatal age 90, probably because of the animals are also doing more activity. There were no differences in food intake, in epidermal adipose tissue and metabolites. We also found, when we studied these animals in the locomotion activity, that enriched environment in a small litter, as you can see here, decreased the time that animals spend in the outer zone, increased the time in the inner zone, and also decreased the time of defecation. So this means that in a small litter, enriched environment could have an anti-anxiolytic effect. When we analyze these animals in locomotion, in the elevated glutamate test, we found that both normal litter and small litter animals that were exposed to enriched environment spend less time in the closed arm, as you can see here. So this also supports the previous work that enriched environment could have an anti-anxiolytic effect. And finally, when we analyze episodic-like memory, we also found that in small litter, if you can see here, so as I mentioned before, a small litter in a standard environment don't have preferences for any object. But when we analyze these animals in this test after the enrichment, we see that animal could distinguish between the object and they recover the temporal component. So they prefer to spend more time with the old object with respect to the recent object, but not the spatial component. So we can say that the enriched environment restore in part episodic-like memory in this animal. So act as a really cognitive stimuli. So up to now of this part, we can say that the enriched environment decreased body weight in both normal litter and small litter, but have no effect in food intake and metabolite concentrates and also epidermal adipose tissue. And more important, environmental enrichment attenuate anxiety-like behavior and episodic-like memory impairments in small litter. So after that, we wonder which kind of mechanism could be involved in this effect, what kind of molecular mechanism. And for that, I want to introduce neurostertiogenesis. So neurosteroids are steroids that are synthesized in the brain from cholesterol or another precursors. It has been studied for more than a decade because they have a lot of positive, it has been reported that they have a lot of positive effects in cognition, memory, learning, plasticity, neurotransmitter regulation, anxiety. So in fact, it's also reported that the treatment with neurosteroid like allopregnanolone could recover animals after some stroke or some brain ischemia. So in the brain, we have all the machinery, all the enzymes and proteins that are necessary for the synthesis of this compound. As you can see here, estradiol, progesterone, allopregnanolone. So more important, on at least what is more important for our work, is that neurosteroids are really implicated in cognitive functions. Particularly, it was reported that allopregnanolone, progesterone, and estradiol restore or regulate hippocampal dependent learning. And that is why we are so interested in understand how neurostertiogenesis is in our model, in small litter and in small litter after, in enriched environment, in both kind of conditions. So for that, to study neurostertiogenesis, we start this work. I have some briefly results related with this, but we start the work studying the expression of the stereogenic enzymes, that are the enzymes that are involved in the synthesis of this neurosteroid. In the hippocampus, that is the main area related to memory and learning functions. So for that, we isolate 3 areas of the hippocampus, CA1, CM3, and dentate gyrus. We use a micropunching technique to isolate this area, and we perform RNA extraction to analyze gene expression, and also DNA extraction to analyze transcriptional regulation. When I am talking about transcriptional regulation, we are particularly interested in epigenetic modifications, in analyze if epigenetic modifications could be involved in the regulation of these enzymes in our model. When we are talking about epigenetic modifications, we say that are reversible modifications that impact gene expression without changes in the DNA sequence. There are another changes like protein modifications or DNA methylation. So DNA methylation is one of the epigenetic modifications that is most studied, involves the addition of a methyl group in a cytosine that is followed by a guanosine, so it's a CG group. When this CG group is methylated in the promoter region of a gene, in generally all the transcriptional machinery is not allowed to bind to the sequence and the transcription is blocked, and we say that the gene is silenced. When this promoter is not methylated, in general, the transcription is active. So in general, what we can say is when the DNA is methylated, you expect that the expression decrease or the gene is silenced. So they are inversely proportional. So, well, as I mentioned before, first we studied the expression of all these steroidogenic enzymes in the hippocampus, in the 3 areas of the hippocampus. So we found difference in 3 of them in small litter animals compared to normal litters. One is the aromatase. So if you can see here, first we have animals at postnatal day 21, a small litter, and normal litter, and here animals in postnatal day 90, small litter and normal litter. As if you see in the dentate gyrus, the expression of these enzymes increase in both at 21 at 90 in a small litter if we compare to normal litter. We also found differences in 2 enzymes that are involved in the synthesis of allopregnanolone, 5-alpha reductase in the dentate gyrus. If you see here, you can see that at postnatal day 90, a small litter reduced the expression compared to normal litters, and 3-alpha HSD in the 3 areas, CA1, CA3, and dentate gyrus. If you can see here, at postnatal day 21, the expression of this enzyme decreased in a small litter, but increased in CA3 and dentate gyrus. So maybe this is a compensatory mechanism. But what is important here is that a small litter affect the expression of this enzyme, and this could be related with changes in neurosteroid. We didn't know yet. We need to perform more experiments to prove that, but this was the first step. When we analyze what happened when the animals were exposed to enriched environment, we found that enriched environment in small litter could control 2 enzymes. The first one is the aromatase. As you can see here, we see normal litter and a small litter in a standard environment, and normal litter and a small litter in enriched environment. If we compare a small litter that was increased, we can see that enriched environment decreased the expression and restored to similar level to normal litters. But contrary, in normal litter animals, enriched environment increased the expression of this enzyme. And this was previously reported by our group. We found that enriched environment would increase the expression of this enzyme. We already don't know why is this different effect in normal litter and small litter. We have to do more experiments, but this was really a surprise for us. Also, when we analyzed the 5-alpha reductase, we found in CA3 that in both cases, normal litter and a small litter, enriched environment increased the expression. And here we can see that, as I mentioned before, a small litter decreased the expression of this enzyme compared with normal litter, but enriched environment tend to increase again, so tend to restore the expression. So basically, what we can say here is enriched environment changed the expression of 2 enzymes, the aromatase and the 5-alpha reductase. So we again need to analyze what happened with neurosteroid here. So based on this result, we decided to study if epigenetic mechanism could be involved in the regulation of these changes. For that, we studied 2 promoters that were affected by enriched environment. The 5-alpha reductase 1 was the first. As you can see here, you have the promoter. This promoter is a big CpG island. This means that the CG site frequency is higher, so it's probably methylated to regulate transcription. There are 5 sites, 5 methylation sensitive sites. We found that one of them, the methylation sensitive site 1, was affected by enriched environment and a small litter. So here, if we compare a small litter and normal litter in a standard environmental, we can see that methylation is increased. These results are in dentate gyrus. This is the first result that we have, that I will show here. So if we remember how the expression was, we found that small litter decreased the expression of this enzyme, and there are an increase in methylation that could explain this decrease. But when this animal, a small litter, was exposed to enriched environment, if you can see pink here and blue, the levels of methylation again decrease. So tend to reverse the methylation of the DNA, and this is related here with expression that was increased. Also, we analyzed the aromatase. So here is the promoter of the aromatase that is reported. We have a 1-methylation sensitive site that is related with a CAMP sequence, control sequence. This sequence is involved in the transcription of this enzyme. And so what we see here is that a small liter reduced the methylation compared to normal liter. And as just to remind you that the expression was increased. But when small liter were exposed to enriched environment, the methylation increased, and is related with a decrease in the expression. So maybe it's complicated to understand because but really complex to understand methylation. But basically, what we found is that enriched environment, I mean, a small liter changed methylation parameters of the promoter of these two enzymes, and enriched environments try to restore the methylation levels and restore the expression. So as a summary, we can say that small liter, the reduction of the liter at postnatal day 21 has an effect in metabolism and also in the expression of steroidogenic enzymes. At postnatal day 90, the effect in metabolism are reversed, but we also see the changes at molecular level, and the changes in this enzyme is regulated. It could be regulated by methylation mechanism, and we also found in these animals an impact in episodic-like memory and anxiety-like behavior. What is important here is one of our hypotheses is that changes in these enzymes could be associated with changes in neurosteroid, and both allopregnanolone and estradiol are involved in the regulation of memory and anxiety. Of course, this is just a hypothesis, and we have to prove that. We need more experiments for that. When these animals were exposed to enriched environment, we reversed the effect at molecular levels, and also restore or attenuate the effect in episodic-like memory and anxiety-like behavior. So as a final conclusion, we can say that this liter reduction has a long-lasting effect on neurostertiogenesis and behavior. So it's really important because show how a nutritional experience or an environmental behavior, environmental intervention during early postnatal development would have effects in a long term in adult animals. But the good news is that effect of liter size reduction could be reversed through environmental intervention. And this is just to invite you, if you want to know more about our models, to our two posters from the lab. One is from Rossetti, a PhD student, is the poster 44, and she will be with the poster this afternoon. And also the poster for another PhD student, Pamela Fernandez, that is the poster 21. So feel free to come and visit us. And as I just want to say thank you again to Guillermina for this invitation, to the Sociedad Argentina of Neurostertiogenesis for allowing us to be part of this symposium, this congress, also to the audience for being here. And of course, the most important people, that is all the people that work with me. This is a teamwork, so I am so proud of them. And of course, we are more than equipment, we are a family. So, and I also want to thank to the institution that fund all this work. Thank you so much.
Well, it's time to questions.
Hi, Flor. Thanks for the very nice talk, Rodrigo here. I was wondering whether you looked at, perhaps I missed this, the restorative effect of the enriched environment. Does it also induce changes in behavior in terms of exploration? And I was wondering if you looked at whether the restorative effect comes from a cognitive process due to the enriched environment or due to perhaps changes in locomotion, exploration, and so on.
Yeah, we really don't know, but we saw that because the time on exploration change of the animals, particularly in locomotion activity, we can see that. And we think that it could be due to motor abilities that, of course, the animals without this kind of environment, that animal don't have. So these animals in rich environment are in constant movement, but we cannot say it's specifically related to motor abilities, but that was one of our hypotheses, that is related to motor abilities.
Thanks.
Hi, Flora. Thank you for your nice talk. This is Marta. In relation to what Rodrigo was just asking you, I guess you don't know it, but this methylation changes, do you imagine that they're going to maintain stable in time? And if these changes might, the second part, might change their feeding behaviors?
Yes, I mean, it could be because methylation change could be along, could accompany all to the animals or along all the life or not, because are reversible. That is a good thing. So that when the methylation change is established, could maintain, and they could affect feeding, of course, particularly because they could also affect changes in other molecules or genes that are related with feeding. There are the 2 PhD students that I mentioned are working in the effect of a small litter and enriched environment more on for feeding. So they are studying reward system and homeostatic system, and also found interesting change in methylation. So of course it could be related with that. I mean, this work was more related with memory effect, but definitely it could be related. Yeah.
Another question. If you can speak in Spanish, the 3 talks speak Spanish if you want. Hablar en español, si quieren hacer preguntas, somos poquitos. Thank you for staying here a Sunday morning, early morning. Flor, thank you, Flor, for this presentation.
Welcome.
Well, in second place, I have the honor of presenting Doctora Gisela Lazarino. She is young investigator at the Center of Social and Affective Neuroscience in the Department of Biomedical and Clinical Science in Linköping, Sweden. She will tell us about the effect of high-fat diet on microglia activation and cognitive function. Well, it's a pleasure see you, Gisela. It's a friend. And if you can share your screen.
Can you see it now?
Yes, incomplete. Yes, perfect.
Yes. Okay. Hi, thank you, Guillermina. It's a pleasure for me to be here at the Sun after several years of not being able to be, like, as well this time I'm not able to be in person, but it's super nice to be able to participate. So I will present a little bit of a project that I'm doing together with some other people. That it's a collaboration that we have in between Sweden and Brazil. So this is a project that we are kind of that started last year. So these are kind of new results for us. Hope you enjoy. So, as you may know, and as Florencia was saying, like overweight and obesity are one of the most important public health programs worldwide, and a key driver of this may be the high and increasing consumptions of high-fat diets, mainly composed by ultra-processed food, together with a sedentary life. And this has been associated with the adverse health outcome like type 2 diabetes, heart disease, stroke, among others. At a brain level, obesity is associated with a loss of cognitive function and an increased risk of developing neurodegenerative diseases and dementia. like dementia and Alzheimer's disease, with evidence from both human and animal research. Many studies have reported that in the hippocampus, high-fat diet can alter the morphology and the structure of synapses, the level of neurotransmitters, and the synaptic plasticity. Sorry, please tell me if I'm going too fast. Besides, high-fat diet worsen age-related cognitive decline, showing highly impaired hippocampal inflammation and cognitive function. And it could be an indication of neurodegeneration. But how fast is the brain affected by high-fat diet? The injection of high-fat diet causes overproduction of circulating free fatty acids and systemic inflammation. Immune cells, free fatty acids, and circulating cytokines reach the hypothalamus and initiate a local inflammation through microglia proliferation. Microglia is the brain resident immune cells in the brain, yeah. So the hypothalamus is the first brain structure reported to be affected by high-fat diet, and that only that one day of diet can alter the gene and the protein expression of pro-inflammatory cytokines and the astrocyte and microglia markers. And 3 days of diet can increase microglia number and activation. So we know that the injection of high-fat diet increases the microglial activity as it leads to a systemic inflammatory response, and all this can lead to neuronal damage. But is the hippocampus affected in the same way? Several articles have reported that rodents exposed to high-fat diet show obesity, cognitive impairment, and increase in microglial activity, and as well changes in microglia morphology. But most of these studies were done in long term or has used 60% of fat in their diets, what is not really representative of the real consumption of a kind of a normal person in our kind of societies. So recently, some Brazilian researchers that actually, some of them collaborate with us, show that only 1 week of high-fat diet can affect the performance of the animals in memory tests, and as well, like a lot of, changes at the body level were found, but they don't report anything about microglia. So could be microglia have a role in this. So for that, in our work, we aim to investigate the onset of hippocampal dysfunction after high-fat diet intake, and which is the role of microglia in triggering this status. Our hypothesis is that a short-term intake of high-fat diet induces microglia activation and neuroinflammation, leading to hippocampal synaptic function and behavioral impairments related to cognition. So, to analyze when the neuroinflammatory processes involving hippocampal dysfunction after high-fat diet intake start and their relationship with behavioral changes, we have taken wild-type adult male mice adult, and we fed them either with standard chow or with high-fat diet with 50% content of 45% content of fat. And we keep them on a diet during 3 or 10 days. 2 days before the sacrifice, we perform an open field test, like to see locomotion, as Florencia explained short before. And 1 day before the sacrifice, we perform a novel object recognition test or an object location test, an object relocation, in order to evaluate working and spatial memory. For this, the animals were exposed to 2 objects that are the same for 5 minutes, and 1 hour after they are tested. In the case of the novel object recognition, we change 1 object for another one, for a second one that is different and that are unknown for the animal, or in the case of the object relocation or object location, in this case, we use a third object different from the other two, and we locate it in another position. So we expect the animal to explore more the object that is new or the object that is relocated, the object that is located in a new position. And well, after the sacrifice, that is the next day of this test, we sacrifice the animals and we take the brains and the white adipose tissue. So the first result that we obtained were like about the metabolic parameters. We have seen that these animals increase the body weight from day 6 of diet. Like in 3 days, we don't see much. There is an increase, but it's not enough, leading to a kind of overweight state by day 10. And this has seen as well at the adiposity index, as these animals increase, and after 10 days of diet, they increase their adiposity index. Regarding the behavioral analysis, these animals didn't change their locomotion, as they perform mostly the same in the open field test. But regarding memory, we have seen that after 10 days of diet, the animal decreased the time spent with the object that was relocated. And as well, we see a tendency, but it's not statistically significant, of the time spent with the new object. Going to the specifically, so then we wanted to see, this means that there could be like change at the hippocampal level. So then we analyze, okay, is there any differences at the microglia of this hippocampus? This has been done in CA1 of the hippocampus study. There is any change in the microglia. So then for this, we have done staining for IBA1, that is a marker for microglia and macrophages, and C68, that is it's a marker for active microglia. You can measure phagocytic activity of microglia by using this one. And we have seen, I hope you can see it properly in the screen, that when we did the counting of the IBA1 staining, so the amount of microglia, we have seen that there is no changes after 3 days of diet, but there is a tendency of an increase after 10 days of diet. And as well, it's not reported here, but the microglia as well co-express a lot with the CD68. So it means that it was active microglia. So, but then we wanted to go further in the study of the 3 days animals. We left them here because we say, okay, there are no changes. We didn't see anything at the metabolic level, no changes at the behavioral level. Let's keep them on pause, sort of say. So we grab the 10 days animals and we run what is called Azure Analysis. I don't know if you are familiarized with this, but it's a measure of complexity and the distribution of the microglia processes. So for this, we take 40x pictures at the confocal microscope, and we inside the row we select like 5 to 7, 8 microglia per animal, and we do this bilateral as well, and we skeletonize them in ImageJ, and we count the number of branches, and then we run a plugin that is called Shoal Analysis, where it shows, like as you can see here, a circle all around the microglia. So the core is in the nucleus of the cell in the microglia, and then all around you have this circularity. So each dot is equivalent to one intersection, what is called one intersection value. So one point of the microglia, any kind of these branches, when it touch one of the circles, it's counted as a dot. So what we do with this information, we generate a graph in which in the y-axis we count the number of intersections on each radius, and then in the x-axis we count the radial distance from the soma of the cell. So as you can see here, we expect this kind of pattern for resting microglia, But if the microglia is reactive, we expect a lower number of intersections. So you see that it's more plain, the graph. So we ran this for our control, for our high-fat diet animals in 10 days. And we have, here you can see how we choose the microglia. It's pretty difficult because we have to do a set stack of all our sections and then check that the microglia are complete and are not cut and so on. So it's not a very, this is very time consuming. That's why we didn't, we need to do it in 3 days. We didn't do it yet. And we have seen, as you can see here, that there are fewer intersections closer to a nucleus. So it means that it's like the cells have a bigger volume, like the cell branches. So we can state that, or we think that in this case, the microglia is more reactive, as is like less complex, sort of say. You can see it kind of here. And then we have done a staining as well in 3 and in 10 days of GFAP to distinguish astrocytes, and we didn't see any. We wanted to distinguish astrocytes from other glial cells, but we couldn't see any difference. Any of this data is published. As I told you, this is kind of a work that we started no long time ago. So then, as it was reported that the high-fat diet leads to an inflammatory state, we wanted to analyze the inflammatory and microglia markers via qPCR analysis. For this, we use the whole hippocampus of the animals. We extracted it we dissected from their brains during the surgery in the sacrifice, but we didn't find any difference in the expression in any of the interleukins that we studied or the microglia markers. Well, it's pretty like weird, but it's still a result. Well, then to change for our second objective, to determine if this microglial activation is necessary for the high-fat diet-associated cognitive decline, and whether the inactivation of the microglia prevent the damage caused by high-fat diet, we examine if DREAD-based inhibition of microglia at a general central level would reduce the behavioral impairments induced by high-fat diet consumption. For these mice that express the inhibitory DREAD, HM4D I, in microglia, specifically, we are used, as you can see here, this is a line. And we use all the CRIPLAS animals as our control animals. So these animals express the inhibitory DREAD in microglia under the CRIP promoter. So I don't know if you are familiarized with DREADs, like I didn't say the designer receptor exclusively activated by designer drugs, but these animals, what you can do is like to inject tamoxifen that makes the CRI signal to express, and that will cut and flip the DREAD signal in the gene, in the transgene, and then when, so it will start to express the DREAD. And for this DREAD to be active, sort of say, or in this case, inactive because is this contract, is the HM, you have to inject CNO. And when you inject CNO, this DREAD becomes active, allowing us to inhibit the microglia expression. So to do this study, we inject tamoxifen for 5 consecutive days. In these mice, in this case, we use male and female mice because the line is not very productive, and we lose a lot of animals. We waste a lot of animals if we don't use both. And well, after 1 month of the tamoxifen injections, we start the diets, standard CHO or high-fat diet. So we have like 4 different groups depending on the type of diet and the type of animals that we are using. And we inject for 10 consecutive days CNO to start the inhibition of microglia. So this started together, the diet and the injection. So some days, like 3 days before the sacrifice, we perform an open field test or a Y-maze test. That is another memory test. And then the day after, we perform the novel object recognition. And one day before the sacrifice, the object relocation or object location. That is the same that I presented before. And as well, this time we control the body weight of the animals. And after sacrifice, we extract again the brains and the adiposity and the white adipose tissue. So when we analyze the metabolic parameters of these animals, we can see here that these are the high fat without inhibition of microglia, the regular ones. And we can see here that the inhibition of microglia decrease the body weight of the animals. So these animals were, here I didn't show the intake of these animals, I'm sorry. Like I just realized that I missed the graph. But these animals tend to have a little increase intake of high fat diet, but not as much as these ones. But still their body weights are normal. As you can see here, the body weight gain was a little bit higher than the control animals or the control diet animals here, but not as much, not significant either. And these animals didn't increase as much the white adipose tissues. When we analyze the behavior of these animals, we can see again that they didn't change their locomotion according to the other groups. But in the novel object recognition and in the object relocation test, they perform better than the high fat diet fed mice. So the inhibition of microglia seems to help in this, or seems to be, microglia seems to be involved in this kind of inflammation or neurodegeneration that causes the animals to not perform well in the memory behavioral test done by high fat diet. And the same we have seen less percentage of alternations in between the arms of the Y-maze by the inhibited microglia animals compared to the high fat diet ones. When we analyze microglia, in this case, we don't have the time yet to do the shoal analysis. Now we have students that it's started to run in it. But as you can see here, the microglia of the high fat diet alone, if you check the amount of C68 that they express, you can easily see, or maybe not easily here, but I hope at least you can see something, that they express much less C68 compared to a high fat diet, the high fat diet with inhibited microglia, and they are almost in their normal levels. And I didn't show here the amount of microglia, but as well as expected, it is decreased the amount of IBA1 reactivity. But sorry that I didn't put the graph here. So what we can conclude, that in our model, 3 days of high fat diet were not enough to increase body weight, to alter memory task performance, or to activate microglia in CA1. But 10 days of high fat diet increase body weight and adiposity, impair the spatial memory and the recognition memory, and increase microglial activity in CA1. And all these changes that have been seen in 10 days were reverted by chemogenetic inhibition of microglia, what could indicate that microglia activation is necessary for the cognitive decline. But one thing that needs to be said here is that we did the inhibition of microglia at a general central level. So our next step will be to analyze the inactivation specifically at the hippocampus. We aim to reach the CA1 in the hippocampus. So that's our next step that we hope be able to do so. And well, thank you for listening. And as I told you, this work is not only mine. This work is a collaboration in between Brazil and Sweden. And I want to thank especially to Priscilla, Mateus, Mira, and Andresa that are the main, that together with me, we are the ones doing this research. And well, David is my supervisor, so he's the one leading the full group. So thank you very much and hope you enjoy.
Well, thank you, Gisela. It's time to question if you want.
Hi, very nice talk. I have a question of like a technical question. How do you calculate the index for the novel object and the novel relocation? Because it was I saw in every graph that the control mice didn't reach the 50% that you dashed. So it means that they didn't prefer the novel object or the relocated object.
Yeah, like that's maybe I can share my screen again to show, or I can answer directly. Yes, our animals actually, in 3 days, our animals didn't reach the minimal level. That's why as well we wanted to keep them. I mean, we continue studying because we have the studies done already, but our animals didn't perform as well. Now these same tests are being performed in Brazil in order to see if they are out, because they bought exactly the same animals as we did, in order to see if their animals perform better than the ones that we bought at the laboratory. So the thing is that the index is calculated by as you asked, the total time that they spend in both objects and divided by the total time that the animal spend with the novel object or the relocated object divided by the total time that the animals spend with both objects. And the thing that we think, something that I didn't say, is that we changed the diet that the animals follow at the animal facility is not the control diet, sort of the pellet, is not exactly the same as our control diet, because then we changed to research diet control diet, that is the one recommended to use for this kind of treatment. So our concern now that we realize is that maybe these animals are as well passing by a change of diet, sort of say, because it's not exactly, the composition is not exactly the same of the control diet that or the diet that we use at the animal facility with the diet from research diet. So that could be as well, I don't know, a confounding factor of the study, that after 10 days, the animals are more stabilized. Now, in the next cohort that we are doing, we change from the very, very beginning to we put them all in the control diet from research diet, and then we start the high fat diet when it's the moment to start. I hope I answer your question.
Perfect.
Hi, thanks for the very nice talk, Rodrigo here. I have to say I'm a total outsider to the field of nutrition, so maybe I'm completely off here, but I wanted to ask about the diet composition and the control diet. So do you keep the overall level of calories constant between the two, like the high fat and the low fat conditions? And if so, do you replace fats by carbohydrates? Or I'm thinking about studies that relate high carbohydrate intake with inflammation and long-term effects like correlations with Alzheimer's, and so whether you looked at like longer-term effects, and how are you replacing calories, or are you just increasing the level of calories with?
No, we are increasing, like I think the caloric content of the diet is little higher in the high-fat diet one. And yes, the calories, I mean, of course, as it has less amount of fat, the diets, the control diet have more amount of carbohydrates. But I think that one thing that as well you have to take into consideration when it's related, as you well said, now, I mean, there are some research done in the amount of carbohydrate and the impact on memory tests, but as well the level of proteins is very important. And in this case, both all the levels of the micronutrients and the proteins is exactly the same. Only like the amount of carbohydrates is changed a little. And of course, we need to take it into consideration as well. It's a very good point, actually.
Hello, Gisela. Carlos from Córdoba. Do you know whether starvation or intermittent fasting could improve the cognitive performance actually in your model?
No, I mean, I didn't found many research that done in that. Probably it is because the main thing is that most of the studies that have been done, it's in very long term of diet, but there is no much done in these short terms. So in the long term, I think long time ago, I saw something about that the fasting were like ameliorating the effects of the high-fat diet. I mean, like ameliorating the effects of the high-fat diet, like this intermittent, but the problem is that as well, sometimes it generates, I don't remember the word in English, sorry, when they eat a lot in a very short term of time when you give back the food. So it's not, I mean, it ameliorates effects in the hippocampus, but not, it's not very good with this diet at least. And eating in a long term in the metabolic levels. But it's something that maybe we can research as well, I think, because it was part of we had it in part of our plan to see if changing the amount of days that we give the diet and increasing the time in starvation of the animals, it decreases the problems at the hippocampus. Microglia. I mean, it decreases the amount of microglia in the hippocampus.
Hi, thank you very much for the talk. I wonder, when you target exclusively the hippocampus with your chemogenetic tool, do you expect to observe some kind of dissociation between cognitive deficits and food intake? For instance, could food intake remain intact?
Yes, I think that, I mean, we think that probably the food intake will not change much. Actually, we were like, now we start to study the hypothalamus of these animals, because one of our kind of hypothesis is that if we target specifically the CA1 of the animals, as the hypothalamus will remain intact, the animals will keep on eating normally. And it is pretty reported as well that this microglia, one of the main actors in the hypothalamus in the short-term intake. So yes, we expect that.
Well, another? Well, thank you very much, Gisela. A great talk. It's a pleasure. Well, finally, I have the honor to introduce to the speaker. Sorry. Well, finally, I have the honor to introduce our third speaker, Dr. Anabolo Garcia. She is an associate researcher at CONICET and work at the Instituto de Salud y Ambiente del Litoral in Santa Fe. She will tell us about her setup of a zebrafish model. Well,
thank you, Guillermina, and good morning to everyone here. I want to thank the organizers for inviting me to this symposium. It is a pleasure for me to be here and have the possibility to talk about this project. It is a very new line of research in our lab and with a new animal model. So it's very nice to be here talking about this. I will talk about sleep that, although have shown to be very important in the control of body weight, it has not received enough attention. Well, the name of my talk is Establishing Zebrafish Dañorerio as a Model to Study the Effect of Sleep Restriction on Food Intake Behavior. Sleep, it's a physiological reversible condition that is regulated by the brain. And although there is not a general consensus about its function, it has been associated to cell repair and memory consolidation. Oh, what happened? Okay, sorry, mistake. Okay, in modern societies, the time of sleep duration has decreased among adults and children during the last several decades. The reasons are, for example, shift works, time schedules. For example, the time children go to school is very early, and also the excessive use of screens, but also the accelerated work pace and the stress associated. This depth of sleep can have small or big effects on our life. For example, it can affect our mood, our alertness. It's very useful very common to feel sleepy after having a bad night, and also affect our cognitive performance, but more critically, it can affect our brain health. Many studies have shown that sleep, decreased time sleep, and also low quality of sleep are risk factors for neurodegenerative diseases. And also, many studies have shown that sleep restriction is a risk factor for metabolic diseases, including hypertension, type 2 diabetes, and obesity. In relation to the, in relation to this, a great body of evidence have shown that sleep restriction, sleep deprivation, or sleep fragmentation in studies, epidemiological studies, but also in control studies in human, produce overfeeding and also increase appetite and the preference for high-calorie food. These overfeeding have been associated to increase ghrelin levels, decrease leptin levels, and the activation of brain areas associated to seeking behavior and the reward effect of the food. As you may know, sleep has been described also in many other animal species, for example, flies, fishes, birds, and rodents. This means that these animals exhibit the main three characteristics of sleep. that are immobility associated to the state of sleep, a low response to an external stimulus, and also the existence of an homeostatic control, which is controlled by neurotransmitters. These animal models can be useful to study sleep restriction. In fact, sorry, I put my finger. Okay. In fact, in studies in rodents have shown similar effects than those observed in humans. Sleep fragmentation, sleep restriction, or deprivation produce an increase in food intake, a reduced energy expenditure, and the associated increase in body weight. There exists some evidence showing that the reduced time of sleep can affect orexin neurons, but there are no studies exploring how food sleep restriction can affect the circuits regulating food intake, the central controls of food intake. Zebrafish has become a very attractive model for research. This is due to the advantages it offers over the mammalian models. For example, the short flight cycle, 3 days after fertilization, you can obtain free swimming larvae, and also after 90 days, you can obtain a mature adult. Other advantages are related to the lower cost and space required to maintain the animals, and also the small adult brain size that permit you to process many samples in less time and less space. And also the availability of genomic databases, tools, and mutants. Zebrafish is considered a good model to study the mechanisms regulating food intake, because it conserves most of the brain areas and functions, and also the most neuropeptides and neurotransmitter involved in food intake control. For example, neuropeptides as MPY, HGRP, orexin, and those involved in the inhibition of food intake as POMC, CART, and dopamine, norepinephrine, and serotonin. Many of these factors are also involved in the homeostatic control of sleep. These are MPY, orexin, dopamine, it's not in blue, but norepinephrine and serotonin. Sleep has been characterized in zebrafish, both in larvae and adults. And it's also conserved the most important factors regulating sleep homeostasis. There are no studies in zebrafish exploring how sleep restriction can affect food intake. And this is our project. We decided to start in this animal model at the institute. It is a new model because most of the people working in our institute work in rodents. And so I will show you the aquarium we started to build about 2 to 3 years ago. And it was a quite challenge to do this because it was everything new, and we started from the very beginning. So the aim of our project was to set up a model of a sleep restriction in zebrafish to study the effect on food intake and also on the factors involved in the mechanisms, the homeostatic mechanisms that are those involved in the energetic homeostasis, and those involved in the non-homeostatic mechanisms that are more related to the seeking behavior, for example, alertness, and the reward effect of food. Well, I will tell you how was the methodology we used. We used, in this case, males adult zebrafish that were localized in the fish tank. We used 2 animals at the same time that were separated by a wall, so they can interact each other, but it was with the purpose to do more animals at the same time only. And the experiment, the ambient light and intensity and temperature was constant across all the experiments. We wanted to produce an alteration in the sleep of the animals, so we wanted to do some disturbances, and we decided to do vibrations inside the water. So for this, we used the alarms of a cell phone. And here I show you the experimental schedule. As you know, zebrafish are diurnal animals. They were entrained to a light-dark photoperiod. The day, the light phase consisted, it was the day, we can say, in 14 hours of light and 10 hours of darkness. At the beginning, they had 3 days of habituation because they were individually located, and this is something maybe stressful for these animals that are very social. After that, they have 2 days with no disturbances at all, that are maybe in pink, yes, a more light color, and then they have 2 days of disturbances in red. Well, we have 3 groups, experimental groups. 2 of them received disturbances. 1 of them received the disturbances during the night. This is the ND group. And the other group, there was a day disturbance group that the purpose of this group was to study how the vibrations by themselves affected the fishes, whether they produce any kind of changes in the activity or stress. And we also have the control group that didn't receive any disturbances during all the experiment. And here we have the schedules of the disturbances during the day. The disturbances started 7 hours after light on, so we have 2 main periods, the pre-disturbances period and the day disturbances period. The disturbances consisted on vibrations that lasted 5 minutes and were active every 20 minutes during a period of 6 hours. These are depicted by the blue arrowhead. During the night, we have the same kind of disturbances that were applied 40 minutes after light off. And in this case, we have 2 main periods, the night disturbances period and the post-disturbances period. We also measure the daily food intake of the animals. We measure during 5 minutes the number of pellets and the milligrams of food they ate. And also the activity of the animal was recorded with a video camera during all the experiment, and we use infrared lights during the dark phase. And this is real life. This is how we recorded the activity of the animals. And on the right, you can see a fish and a pellet food that is on the surface of the water. And here I will show you how we measure the sleep in the animals. We use the software EasyTrack that is an open source because it's very expensive, the software for this kind of measurements. And this permits you to select the area of the screen, and then it shows you a plot that it shows the pixel changes, as you can see there. Then you can set a threshold under which you consider the animal is still quiescent. And the literature have described that a quiescent state of more than 6 seconds is you can consider the animal is asleep. So we define it that the mean duration of this quiescent state over 7 seconds or more to consider the animal were asleep. And here we have the results of motion during the day. We we compare the two periods, the 2 days with no disturbances in orange, I don't know what color is this, and the 2 days of disturbances in red. And as you can see here, we did not observe any changes in the motion of the animals during the day. This was good because in the third group, the DD group, we wanted to see if the disturbances during the day could affect the motion of the animals, of the behavior. And see, we can hear that there was no differences, and this was very good because vibration during the day didn't affect the activity of the animals. Then we analyzed the percentage of sleep during the night. Again, we studied the difference between both periods in the 3 experimental groups. And as you can see here, we saw that the animals that received the disturbances during the night slept less in comparison to the days they did not receive the disturbances. The other 2 groups, the control and the group that received disturbances during the day, did not show any differences. And this is what we wanted to see because we wanted to prove that these vibrations produce wake up the animals during the night. Then we analyzed the time when the animals fell asleep and woke up. We measure in minutes from the light off, and we didn't see any significant differences between the both periods and in any of the 3 groups analyzed. We also did not see any differences in the duration of sleep during the night. And then we wanted to know the effect during the both main periods of the night, the period of disturbances and the post-disturbances periods. And as you can see here, the effect was observed during the disturbances period. This is the first graph, where you can see that during the night, the disturbances produce a reduction of sleep. But after this period, there was no differences in the ND group and neither in the other 2 groups. Then we wanted to know how the vibrations affected sleep. For this, we analyzed the effect of vibration, these 5 minutes of vibrations on sleep. This is in blue, and we compare with the 15 minutes that follows this period in light blue. And as you can see here, during the 5 minutes of the vibrations took place, there was a reduction in the time of sleep, in the percentage of the sleep, and after that, the fishes start to sleep again. And here we have the result of food intake. We compare again both periods in the 3 groups, 3 experimental groups. And as you can see here, the disturbances during the night produce an increase in food intake when they were applied, during the night, but not in the other group where the disturbances were produced during the day. This effect was observed when we measure both the number of pellets and the milligrams of food the animal ate. when we analyze the change in food intake between both periods, we can see that there was a significant increase, almost 30% in the group that received the disturbances during the night, that was significantly different from the other two groups, both in the number of pellets and the milligrams. According to this, as a conclusion, we can say that it constitutes a sleep restriction model in zebrafish that produce an increment of food intake as has been described in mammals. And here we have the analysis we are having in progress. We want to analyze the levels of expression of the neuropeptides involved in the regulation of food intake by real-time PCR, and we are working on that now. And we also want to analyze the neuronal activation in different type of neurons involved in both in control of food intake and sleep, and using some markers of neuronal activation of CFOS, PERC during immunohistochemistry and immunofluorescence. And we also want to complete the study in the females that is doing at the moment. And for future perspectives, we have some funding now to continue the study. And so we want to improve the setup for activity registration, the camera, the infrared illumination that are very important, and also for the activity analysis to obtain more parameters than those obtained here. And we are working, starting a collaboration with the Research Institute for Signals, System and Computational Intelligence. We want to develop a new software to analyze the activity and maybe analyze more than one animals at the same time. That is a big challenge. Well, I want to acknowledge all my group, the neurobiological group at the Instituto de Salud de Ambiente del Litoral, and the people from the institute, and also from the Universidad del Litoral. Thank you for your attention.
Well, it's time to question if you want.
Hello. Very interesting, your work. Thanks for sharing. I just wonder if, do you know if you change the food intake? I mean, either changing the frequency or the composition, trying to connect with the other talks of the symposium. Do you know whether these changes could affect the sleep behavior, like a feed-forward mechanistic loop?
Yes, this is something we want to do. We want to change the kind of food, and also the schedule time of we give the food. And we are planning to do this, but at the moment we haven't done. But thank you for the question.
Hi, over here. We talked yesterday actually, and I forgot to ask you something.
Okay.
Have you considered an enriched environment, or the fact that the fishes are social animals, so being in isolation might affect their food intake? You know,
yes,
anxiety levels or stress?
Yes, thank you for the question. Yes, this is very important. The condition, they are isolated, and this is not the natural condition for these animals. And the problem is that for the analysis of the activity, we need to have individuals, animals, because the software cannot differentiate two animals at the same time. But in relation to the stress, we perform this these groups with the vibrations during the day in order to see how the vibrations affected this. And it could be very interesting to measure cortisol also to see the level of stress. But I have already, I have read some articles showing that it is described that the stress reduces food intake in fishes. but not increase it.
Hola, Nadal. Well, thank you for all the three speakers and the audience. It's a pleasure to stay here, and really thanks to the Sociedad Argentina de Neurociencias for permit that. And yesterday we talked about the possibility to show our results, our topic, and there is a way that our issues state and represented in the society, and it's a pleasure for me, and thanks very much. Go to the coffee break to the Ifibine, and then the last symposium, the number 5.